Hello, this is Icons Team 7. My name is Morgan.
Hi, I'm Habiba.
I'm Sam.
This is Sean.
Hi, I'm Sarah, and this is Let's Get Bacteria Drunk, the experiment. The COVID-19 pandemic has created cause for concern in a multitude of different aspects. As a result of the rapid spread of this new respiratory virus, measures revolving persistent disinfection and sanitization have been implemented into our daily life and have become the new norm. Our group study aims to investigate the effects of ethanol on existing bacteria found on frequently touched surfaces. Antibacterial resistance is a growing concern, and with increased usage of hand sanitizer as a result of the COVID-19 pandemic, it is important to have information that is new and relevant to these circumstances. Additionally, previous studies or notable activists in the scientific community have come to conflicting conclusions on this matter, and therefore there's a desperate need for greater research on the topic since hand sanitizer usage has now become a staple in the daily lives of many. Our research question is, has the tolerance of bacteria to ethanol become prominent in commonly used locations?
In order to conduct this experiment, 10 bacteria samples were taken on different cotton swabs for environmental surfaces and plated on respective agar plates. The samples were incubated at 37 degrees Celsius for 24 hours. Throughout the experiment, photos were taken hourly, excluding the 12-hour night period throughout the 36-hour total period of experimentation. After allowing for the 24-hour initial growth period, one piece of blotter paper was applied to 9 of the dishes with the concentrations of 60%, 70%, or 80% ethanol, and the remaining dish served as a control with no blotter paper. Then, colony growth was analyzed from the photos taken in the immediate 12 hours after the blotter paper with ethanol was applied.
So for each of the 10 trials, we used the pictures of the Petri dishes at 9 a.m., 3 p.m., and 9 p.m., so at 6-hour intervals, and fed them into the software ImageJ. In ImageJ, we edited all the pictures so that they were all cropped to just the Petri dish, then we scaled all the images to be 1200 pixels in length and 1200 pixels in width. After this, we carefully edited the images, leaving only the bacterial colonies as red. So if you look at figure 1, the first image is just a picture we took of the dish, and then the image on the right is the finished edited product, with only the bacterial colonies as red. We then applied a color pixel counter that counted the amount of red pixels in the image. So essentially we counted the area of the bacteria in each image. We then did this for each trial at those 3 times. It's important to mention that in some pictures, the colonies formed underneath the marker writing and were covered by it and thus couldn't be counted by the software. But despite this and some other errors, we still got a pretty good representation of the bacterial colony area through the pixel counter. This numerical area gave us an idea of how much and how fast the bacteria were growing in different ethanol concentrations and how those could be compared. The data was then analyzed by assessing the change in area of bacterial colonies between times to plot the percent increase in bacterial coverage.
Our graph shows bacterial growth in varying concentrations of ethanol. On the y-axis, we have the rate of bacterial growth over the 12-hour period that we analyzed, and on the x-axis, we have the ethanol concentration added to the agar plates. The rate of bacterial growth was quantified by percent increase of colony area per hour, and each point represents the average of the rates of growth of our 3 trials per ethanol concentration. You can see from the graph that there is a decrease in bacterial growth at concentrations of ethanol above 60%. We also see growth in all 3 concentrations of ethanol. This differs from earlier studies that suggested that 70% ethanol would take care of 99.99% of bacterial colonies. Even on the plates with 80% ethanol added, we still see a pretty high growth rate, which could suggest that the bacteria we collected may have ethanol tolerance.
Although the data displayed would make sense in the interest of the topic and align with our hypothesis, we have to acknowledge changes made during the procedures due to limitations of the experiment. From lack of access to certain equipment and inherent experimental design flaws. This include, but is not limited to, small sample size, immature plating techniques that resulted in small breakage of agar plates, and an imprecise method differentiating dead and alive colonies. Hence, the experimental results are inconclusive, but do demonstrate a need for future research. Some possible future research include testing with isopropyl alcohol, exposing bacteria in the same petri dish to increasing concentrations of ethanol, and similar procedure with more sophisticated methods of measuring bacterial growth and dead or alive bacteria.